Oh, good afternoon, everyone, and thank you so much for inviting me to speak this conference. It's great to be here. I've enjoyed all the talks. My name is Dr. Rose Stiffin. I'm at a small private university in Miami, Florida called Florida Memorial University. We don't have a lot of equipment and we don't have a lot of resources, but we are exposing our undergraduate students to research. And so the topic that I'm going to be presenting today is actually a topic that we designed and a student performed. And the topic is green chemistry honey-based synthesis of silver nanoparticles and their evaluation for their antimicrobial activities. The student is Ms. Davianne Forsyth, and this was her senior project, and her co-mentors was myself and Dr. Marilyn Sherman. So if someone could go to the next slide, please. So this is the outline that I'm going to follow, where we will be discussing the importance of the study, the experimental design, and the results. Next slide. So these are some of the key terms. We synthesized nanoparticles using green chemistry. We looked at two different bacteria, gram-negative and gram-positive, mainly because they have a different proteoglycan layer, and we want to see if the nanoparticles have a different way of penetrating the two different bacteria. And we want to explain about a compound called methyl glyoxal and why it is important for antimicrobial activity, and of course that these bacteria have the potential to be pathogenic. Next slide. Next slide. So the importance of the study, and this has been mentioned in previous talks at this conference, there is an antimicrobial resistance, and this is causing a rising crisis worldwide. And so therefore we need to develop new drugs that can attack these superbugs, these drug-resistant microbes. One way to do this is to look at natural resources along with nanotechnology. Next, thank you. So some of the nanomaterials that we're going to be looking at, although there are several, we're going to be looking at the synthesis of nanoparticles using green chemistry. You're moving a little too fast. So the use of nanoparticles, especially metallic nanoparticles, antifungal, we studied, antibacterial. We want to look at gene or drug delivery as a future study, anti-cancer, and of course, antiviral. We are looking currently at antifungal and antimicrobial or antibacterial. Next slide. So how do nanoparticles generally work, especially those that are metallic design, such as silver nitrate nanoparticles? Well, first of all, they can inhibit the proteoglycan layer of a cell, especially a bacterial cell, damaging the cell wall. That kills the cell. They can cause cell membrane damage. They can create reactive oxygen species that can lead to cell death. There's always DNA damage or protein denaturation. And of course, we can inhibit enzymes that are necessary for the cell to live. So in the case of the bacteria, we want to see damage caused by one of these mechanisms of action, especially the generation of reactive oxygen species or to cause cell damage. Next slide. So the three honeys that we looked at to synthesize the silver nitrate nanoparticles were manuka, acacia, and buckwheat. And it has been already studied and known that a lot of honeys are antimicrobial, antifungal, anti-cancer. But we wanted to see if we could synthesize these nanoparticles using honey as a natural basis, therefore green chemistry, to analyze the silver nanoparticles' efficacy in bacterial growth. Next slide. So manuka honey is from the manuka tree. It is a monofloral honey. It produces as a byproduct of glycolysis methyl glyoxal, and methyl glyoxal has been shown to be antifungal and antibacterial. So we were assuming that because of this compound present at high concentration in manuka honey, that with the addition of the nanoparticles, that the manuka honey would be the best antimicrobial against the bacteria when we synthesize the nanoparticles. Next slide. So we have two more honeys, acacia honey, which is from the nectar of acacia trees, and its antimicrobial effect is best at low pH. And buckwheat honey. Buckwheat honey has a high level of phenolic compounds and flavonoids. We already know that flavonoids and phenolics have anti-fungal and antibacterial activities along with anti-cancer. So we definitely want to study all three of these different honeys to see how they could affect the nanoparticles formation and inhibition of bacterial growth. Next slide. So the bacteria that we looked at, the first one, which is a gram-negative one, is Enterobacter cloacae. And this bacteria causes opportunistic infections along with pain. So we definitely wanted to study it because it can become pathogenic. And we wanted to see if this would be a good way to study the use of natural products to inhibit bacterial growth. Next slide. The second one is a gram-positive bacterium, Staphylococcus epidermidis. It is found on the surface of the skin, but it can become opportunistic if it overgrows, causing pathogenic effects such as nosocomial infections, especially on the skin. And we wanted to study that and see how it could be affected by the use of nanoparticles. Next slide. So the purpose of this study was to determine the characteristics of silver nitrate nanoparticles synthesized with the three different types of honeys, and then to evaluate which of these nanoparticles would have the greatest antimicrobial activity. Would it be from the manuka, the acacia, or the buckwheat honey? Next slide. So our hypothesis was that silver nanoparticles that would be synthesized from manuka would have the greatest antimicrobial activity. And we made that assumption based on the fact that manuka has the highest level of the methyl glyoxal, which has been known to be antimicrobial. Next slide. So the materials we obtained from Amazon, we obtained the three different honeys, silver nitrate. We used the Mueller-Hinton agar. And the bacteria, as I said earlier, one was gram-negative, one was gram-positive that we got from Carolina Biological. And our positive control for antimicrobial growth was chloramphenicol. Next slide. So we made our nanoparticles using the honey at different concentrations. The one we worked with was at 50%. We synthesized, we made our solution of silver nitrate. We cultured our bacteria. We prepared our Mueller-Hinton agar for the bacteria to grow. And then we did the antimicrobial testing using the well diffusion assay. Next slide. So in order to do green synthesis, now this is using a plant extract. So I'm going to move on to the next slide since we are going to talk about that later on, but we didn't do plant extracts with this study. Next slide. So this is with the green the green chemistry using honey. We get the metal ions, put them into a solution. We add the honey, and then during the process of stirring and heating, we form the nanoparticles form, and they do the capping and the reducing of the nanoparticles. And so now at the last, on the lower left-hand side, we see the nanoparticles that have been synthesized using the honey and the silver nitrate solution. Next slide. So we did the well diffusion assay where we punched holes in the agar, the Mueller-Hinton agar. We added a certain amount of the material that was made from the nanoparticles as a solution, roughly 150 microliters per well, along with the water as a negative control and the chloramphenicol as a positive control. Next slide. So this is how we know that we synthesize the silver nanoparticles. As you know, a chemical reaction is always indicated by a color change. When we did the stirring and the heating for 30 minutes at 60 degrees, there was a color change for the nanoparticle formation. and we see the 3 different honeys and the 3 different colors for the acacia honey, the manuka honey, and the buckwheat honey. You can see that there's a color change. We do not have a way currently of actually characterizing the formation of the nanoparticles, such as using SEM or UV-vis microscopy. Thank you. Next slide. So this is the Whale diffusion assay, and this is a representative of one of the studies that we did. Now, in the center is our positive control, which of course is chloramphenicol. I don't know if my pointer works, but I'm going to try. This is the positive control. This is the negative control. Of course, we should see no inhibition of growth. But now what I want to call your attention to is the nanoparticles that will form with the acacia honey. The acacia honey is here. There's no zone of inhibition. But when we added the nanoparticles to the acacia honey, there was a significant inhibition of growth, about 50% that of the control. With the manuka honey nanoparticles and the buckwheat nanoparticles, although there was an increase in zone of inhibition, the increase was only about 30% above background. So right now, our hypothesis was proven to be that manuka was not the best honey for the nanoparticle formation or antimicrobial activity. It was the acacia honey. On the right-hand side, we see a similar structure with the other bacterium that we used. And again, acacia honey had no activity, but the acacia honey nanoparticle was about 50% that of the positive control, indicating that when the acacia honey was used to cap and reduce the nanoparticles, it worked better than the manuka honey nanoparticle or the buckwheat nanoparticle. Next slide, please. So now this is simply a bar graph of the data that I just showed you. This is the chloramphenicol. The zone of inhibition in millimeters was about 31, which is significantly high. The acacia honey had absolutely no inhibition. However, when you look at its nanoparticle, it is about 17.1 millimeters, a little bit over half of that for the positive control. Although the manuka honey and the buckwheat honey did show an increase in inhibition when we added the nanoparticles, the increase was not as significant as that of the acacia honey for the Staphylococcus epidermidis. So that means that the acacia honey is working better than the other 2 honeys when we form the nanoparticles. Next slide. We see a very similar treatment with the E-cloque, the nanoparticles from the acacia honey was about 50% that of the chloramphenicol, which is a positive control. Acacia honey itself had no biological activity. The buckwheat and the acacia and the manuka honey did show background activity, and the nanoparticle activity was just 50% above the background, indicating that although they did show an increase in antimicrobial activity with the addition of the nanoparticles, they were not as effective as the nanoparticles formed from the acacia honey. Next slide. So we also prepared the honey-based nanoparticles for SEM analysis. However, our results were inconclusive, and I would love to collaborate with someone who can work with us. We are definitely going to keep pursuing the synthesis using green chemistry as a mode of synthesizing nanoparticles of different metallic structures. And we want to characterize them using SEM for morphology and size. Next slide. Now, this is a deviation from what I just presented. As I said, we have been focusing on nanoparticle synthesis using green chemistry. This is the results of another project that a student did in which she used copper sulfate as her metallic salt, and her plant was the Cedar acuta, and she did an extract using aqueous media. And then she synthesized her copper sulfate nanoparticles using the plant extract. And the plant extract had the components to allow for reducing and capping of the copper sulfate nanoparticles. And then she synthesized the nanoparticles at pH of 7 for 30 minutes at 60 degrees stirring and heating. Next slide, please. So she looked at E. coli, which is a gram-negative bacterium, and she saw that the nanoparticles did have antimicrobial activity as compared to the positive control. However, copper sulfate had a significant background, even though the nanoparticles formed with the plant extract had a higher inhibition assay. We just know that we need to maximize copper sulfate's ability to work as an antimicrobial when it is placed inside of a plant extract as a nanoparticle. Next slide. We also looked at S. epidermidis, which we looked at earlier with the honey, and the copper sulfate did work, but the nanoparticles worked slightly better, but there was no significant difference. We want to do a further study looking at perhaps pH, temperature, and stir time for the nanoparticle formation to see if the copper sulfate's nanoparticles can be enhanced using this plant extract. Next slide, please. So some of the discussion points, and Ewan et al. Said in 2022 that we need to develop more novel compounds that can be used as antibiotics, because currently there are microbes that cannot be killed in typical ways, and also there's a misuse of antibiotics and other antibacterial agents. Boozer et al. Said in 2020 that honeys do possess antimicrobial activity, and that they may vary depending on the floral source and their processing. But we also looked at the use of different honeys to synthesize our nanoparticles. Lastly, in 2021, it was said that nanoparticles can be used with great potential benefits, and we need to make sure that we look at any risks involved, because we know that nanoparticles can be used for drug delivery, for anti-cancer and antiviral therapy. Next slide. So these are our conclusions. We see that acacia honey had the highest increase for the nanoparticles as a way to inhibit bacterial growth. Because we did green chemistry, this is a environmentally friendly way to synthesize nanoparticles. We use water as our solvent. That meant that we created or used no harsh chemicals, and we used honey. And because we use honey, that means that we are using natural resources. Now, we do need to characterize our nanoparticles using some type of microscopy or spectroscopy. Currently, we do not have the resources to do that. Next slide. Next slide. So what are our future studies? We want to do some SEM, and I am going to contact Dr. Gomez in Mexico to see if he can help us out with that. We want to keep doing comparisons of nanoparticles for antifungal, anti-cancer, and antioxidant activity. Not just using silver nanoparticles, we've also used sodium selenite, and sodium selenite nanoparticles worked very well in acacia honey. And we want to look at other metal nanoparticles, such as maybe palladium or gold, using different honeys other than the ones we have. We're currently looking at lavender honey as an antimicrobial, but also to synthesize nanoparticles. And we want to see if we could use these nanoparticles that are synthesized in honey as a way to deliver drugs for cancer research. Next slide. So we want to acknowledge this conference and to allow us to present our research. Next slide. These are our references. Next slide. So if you have any questions, I'll be happy to entertain them. My name is Dr. Rose Stiffin. I'm the chair of Health and Natural Sciences at Florida Memorial University. That was said on the first slide. I would love to hear from anyone who has any questions or suggestions about the